Hello, again, this is Professor George Brooks from the Department of Integrative Biology and Chair of the Center for Research and Education on Aging at UC Berkeley. Earlier, it was my pleasure to welcome you to today's meetings. And now it's my distinct honor and pleasure to introduce today's lecturer, Dr. Judy Campisi, who will also receive the Paula Timmerus Memorial Lecture Award. Just briefly about CREA again. CREA's mission is to investigate the basic processes that cause aging with the goal of improving and extending human health span. And to do that, we work with a number of different cellular and molecular techniques. We use structural biology, and not only that, we collaborate with colleagues on campus who can set the social and psychological networks in which we age. So in terms of introducing today's lecture, first a bit about Professor Paula Timmerus, who is co-founder and the driving force behind CREA. Paula Timmerus received her MD degree from the University of Rome in 1947 and her PhD from the University of Montreal in 1953. She joined the Berkeley faculty in 1958 and served as chair of Physiology and Anatomy Department 1978 to 1984. She was author, that is, Dr. Timmerus was author of 15 books and more than 400 articles in Italian, French, and English. Her native tongue actually was French, even though everybody thought that she was Italian because of her fluency in that language. Her lasting legacy is a massive book she edited, which is called The Physiological Basis of Aging and Geriatrics. It's a classic in the field. With that introduction, it's my pleasure to introduce the president and present Dr. Judith Campisi, who is a recipient of the 2022 Timmerus Award. Dr. Campisi is the fourth Timmerus Award recipient, previous recipients being Elizabeth Blackburn, Marion Diamond, and Cynthia Kenyon. Importantly, Judy was a co-founder of CREA. Judy Campisi received her PhD in biochemistry from the State University of New York at Stony Brook. She completed postdoctoral training in Harvard Medical School and joined the Boston University School of Medicine shortly thereafter. She joined the Lawrence Berkeley National Laboratory as a senior research scientist in 1991. She established a second laboratory soon thereafter at the Buck Institute in 2002. And while not traveling to national and international meetings or being at USC, Dr. Campisi carries on a very diverse research program at the Buck Institute for Research on Aging. Briefly, Judy's research is on the causes and mechanisms of cellular senescence. Practically, this means that her work is highly relevant to several fields, including aging and cancer. She's author of several hundred publications herself, and she's also authored or co-authored numerous book chapters. At the Buck Institute, Dr. Campisi directs a large, multidisciplinary, multi-institutional research training program or programs funded by multiple sources, including the NIH. Dr. Campisi has received international recognition for her contributions to the understanding of aging as being the single largest risk factor in developing a whole wide range of diseases. Judy, I'd like to break tradition now, and instead of presenting you with a piece of art glass, we at CREA would like to present you with this embossed vase. We hope that you will treasure this as we treasure the work you have done in aging and cancer research. And occasionally you might find time to put a bouquet in to not only remember your lecture today and being a recipient of the Timmerus Award, but also to remember a bit about the life of Paula Timmerus. Judy, welcome to the podium. It's my honor to introduce you. Please, we'd love to hear your lecture. Thank you very much.
So let me start by thanking George for inviting me to give this talk and the committee that awarded me the Tamiris Award. I was a huge fan of Paulo Tamiris. Well, many of us were. And it really brings tears to my eyes to think about her and all the work she did for aging research here in the Bay Area. And so thank you very much, all of you. So it's my pleasure today to talk to you about cells that are called senescent cells, and how they impact aging, and how they might be a little window into how to develop anti-aging drugs. So we all know that there's lots of stressors in our lives. This is really what drives aging, is these different stressors. Many of them have been evolutionarily selected, meaning that metabolism, which we need in order to survive, have an impact on our stress level. The environment, of course, does, and we have to live in an environment. And many of these stressors and environmental insults will cause damage to the DNA and often mutations. What that results in is a cellular response. And the cellular response can be myriad. So one is this process called cellular senescence, which I'll tell you about in a moment. But it can also cause cells to die. It can cause cells to enter a state called dormancy, in which they remain sort of poised in time and place and don't really do very much until they get a special stimulus. And of course, what those mutations can do is they can cause cancer. And that is really the primary goal of senescent cells, is to stop the development of cancer in young organisms. So how are these states determined in the face of these different stimuli that we're exposed to? And also, what determines whether these cell fates change in vivo in our bodies? So let's start with cellular senescence. So this is an arrest of cell proliferation. It's a change in phenotype. Cells begin secreting molecules that alter neighboring cells, and it's nearly static, meaning it doesn't change very much over time, so far as we know. I should point out, these are still early days, and so we may change our mind in the next year or so. Then there's cell death. That, of course, is completely static. Once a cell dies, it's gone. Dormancy, as I said, is changeable. Cells enter this arrested state where they're kind of in no man's land, but they can change. And then, of course, there is oncogenic transformation, or cancer, which is highly variable and changes even as a tumor is developing. So what all of these cell fates require is a stimulus. Something needs to drive a cell into opting for one of these states. But it also needs a permissive tissue or systemic environment that allows the cell to express that particular cell fate. So cellular senescence is particularly interesting because it's what we call a tripartite phenotype, and it's under heavy evolutionary pressure, because the main thing that happens is that cells stop dividing. And when a cell stops dividing, it cannot form a tumor. And that's primarily the evolutionary driving force behind this cell fate. But in addition to no longer being able to divide, and therefore no longer being able to give rise to cancer, the cell develops a very complex, multifaceted secretory phenotype. We call it the senescence-associated secretory phenotype, or SASP. And it is changeable and it varies a lot. And I'll show you that it can have profound effects on neighboring cells. And then the cells tend to be somewhat resistant to undergoing cell death. They do die, we can make them die, but they are actually more resistant to cell death than, say, other types of cell fates. And so a way to think about senescence is it's an evolutionary balancing act, because the secretory phenotype is not all bad. It actually helps things like wound healing and tissue repair. And so we think of this phenotype as something that actually can balance the good and the bad versus when stimuli are subjected to a cell. So the main problem that we face as biologists is that there are very few, if any, really specific biomarkers for each of these cell identities. So we have to use a list or a conglomerate of biomarkers in order to say there's a pretty good chance that this cell is in some way senescent or dormant or cancerous. I think when a cell is dead, that's the one thing that we can be sure of. And so we also need to rethink, we biologists have to begin rethinking how we actually go about identifying cells. So if you think about the secretory phenotype, I told you that it's very complex. It can definitely affect neighboring cells, in some ways good, meaning helping them repair tissue, in some ways bad. In that sense, it can induce inflammation, which we know is a major problem during aging. And so the question is, how do we combat these cell fates? And there are basically two major strategies that are being used now to try to control this cell fate. One is to cause the cells to stop secreting all this stuff. And the other is to actually cause the cells to die, to force them to overcome their resistance to cell death and to die. So we have tried both. We're a little bit more in favor of the latter one, because if you think about it, since senescent cells accumulate slowly during aging, and they're somewhat rare, they're not like there's tons of cells in your body that are senescent, then you can use a drug that requires only intermittent dosing. And indeed, there are some drugs that have been and are being developed that will cause senescent cells to die. So this is the approach we took. We made a mouse model. Now, we all know that mice are not humans and they're not perfect with regard to reporting what humans do, but they're pretty good. And this mouse, we think, was pretty good. So what we did was we took a gene that is known to be expressed by many, although not all, senescent cells, and we engineered that gene so that it turned on a reporter that is a molecule that enables us to see the cells in vivo with luciferase, to sort the cells from tissue with a fluorescent protein, and then this killer gene. This is a viral thymidine kinase, and it takes this prodrug, ganciclovir, and turns it into a very potent DNA chain terminator, which is lethal to all cells. And what we're able to show is that although senescent cells don't replicate nuclear DNA, because they don't divide, they do replicate mitochondrial DNA. And so this modified toxin goes into the mitochondria, fragments the mitochondrial genome, and the cells die by apoptosis. So we have shared our mouse with literally dozens of laboratories. Actually, there's another mouse that was made by our colleagues at the Mayo Clinic that's similar. It has the same P16 promoter, the same gene driving, but a different killer. Between the two of us, we have disseminated our mice across a very large part of all of biology that's interested in human age-related diseases. Now, what I've listed here is a partial list. I've actually given up trying to keep track of all the diseases in which eliminating senescent cells has been shown to either prevent the disease from happening, slow the disease down from developing, or in some cases, even reverse the disease. And so it depends on the tissue, it depends on the disease. Whoops, sorry. So this is really good news. We have a transgene that can act on multiple age-related diseases. And what we'd love to be able to do is to find a drug that can do what our transgene can do. So the good news is these drugs are being developed. There are two types. The first is called senolytics, and these are drugs that are designed to selectively kill senescent cells. There are some that are now being used in a clinical trial, they have moderate success. I'll point out some of the problems with all of these drugs so far, is that the selectivity is not as good as we would like. So that's something we all need to work on. The second class are called xenomorphics, and those are drugs that basically tell the cell to stop secreting. So you shut off all of those CES molecules that the cells are making. And what has happened is that if you use these drugs, you can usually have some effect on the presentation of these age-related diseases. Now, here's the problem though. If you want to know whether a drug is working in a human, say to alleviate osteoarthritis, so you give a patient a drug and you monitor their osteoarthritis. It takes years before you begin to see real health benefits. And so the question then is, how can we monitor how good these drugs are without waiting for years for there to be a real difference in health span? This is something we're all working on now, is to try to find better ways to monitor these drugs and to understand when we can tell they're working. Obviously, what we need are good biomarkers. This is something that many labs are working on, including ours. And we need better biomarkers in order to be able to better monitor whether or not these drugs are working. So I just would like to thank you all for your patience, to George and the whole group for having me here, and mostly my entire lab, which is wonderful, as well as all our collaborators without whom this work would not have been possible. Thank you.